Right. Hello, everyone, and welcome. My name's Robin, account manager at Specialty Chemicals magazine, and I'm delighted to introduce today's webinar. We're thrilled to be joined by two leading companies in the field, Chemeda and Essensis, who will be hosting today's session on deuteration reagents for pharmaceutical advancement. Our expert hosts are Dr. Carl Matos, Technical Director at Essensis, and Dr. Catherine Burns, Life Sciences Sales Manager at Chemeda. Thank you for joining us. And with that, I'll hand over to our speakers. Enjoy the session.
Good afternoon, everyone, and thank you for taking the time out of your day to attend this webinar. I believe you will find it interesting, informative, and thought-provoking. My name is Catherine Burns, and I am a Life Science Sales Manager at Chemeda, and I am joined for this webinar today by the esteemed Dr. Carl Matos, Technical Director at Essensis Specialities. And to be clear from the outset, Carl is the man with all the answers. Firstly, I'd like to introduce Chemeda and Essensis Specialities for those who may not be familiar with us. Chemeda are a specialty raw material source and supply company, headquartered in Ireland, but a global company with customers and suppliers all over the world. Founded in 1988, we are now a team of 60 people, and we've about 300 customers and more than 400 suppliers worldwide. As a company, we market a select range of specialized chemicals and ingredients to the marketplace. But in addition to this, we also offer our sourcing expertise to find the appropriate solution to our customers' raw material supply needs and challenges. Our tagline: source, supply, solution. Essensis Specialities are a principal partner of ours within the Life Science division, and their commitment to innovation and excellence perfectly aligns with our vision at Chemeda. We've worked with Essensis Specialities for many years, in particular with the Evans City site for over 30 years, and we represent them as their distributor in Ireland and the UK. Essensis Specialities is a global leader in catalysts, ligands, building blocks, and specialty reagents. Their product portfolio includes alkali metals, boranes, borohydrides, specialty alkalates, peptide synthesis reagents, water-soluble phosphines, and high-purity metal salts, but to name a few. Essensis Specialities operate 3 sites in the US and 1 in the UK. In the US, they have a hydroelectric facility in Washington State, a Pennsylvania plant that's been in operation for more than 80 years, and a Massachusetts facility that handles laboratory, high pressure, and cGMP manufacturing. Their UK facility in Cambridge offers aromatic building blocks for APIs, as well as custom manufacturing solutions, and in particular, long-chain aliphatic halogenated compounds for the life science industry. Essensis Specialities' ethos can be summarized in the following line from their website. We challenge ourselves to develop chemistries that simplify, advance, and optimize life in every way. So that brings us to today. The pharmaceutical industry is constantly seeking innovative ways to develop safer and more effective drugs. One such innovation that has gained significant attention in recent years is the area of drug deuteration. This webinar explores how advanced deuteration reagents are driving innovation in drug discovery and development. Carl is going to discuss deuteration and its impact on API development, including the challenges and the opportunities this presents. We'll also explore real-world applications, and Carl will illustrate the selectivity and reactivity of the Essensis deuterated reagents. Before I hand you over to Carl, I'm just going to put up a survey that I would ask you all to have a quick look at. We're trying to gauge how familiar you are with the use of deuteration reagents in pharmaceutical development. There are 4 options. A, very familiar. I've used or evaluated them in my work. B, somewhat familiar. I know the basic concept, but not the details. C, heard of them, but I'm not sure how they're applied in drug development. And D, not familiar at all. This is new to me. I would encourage you all to answer the survey, as it gives us a better understanding of the group here today. We'll run a second survey later in the webinar, and again, I would ask that you get involved. It's also important to mention that you can put any questions in the chat section on your screen as we go through the webinar, and we'll take time at the end to address any questions from the room. And now, over to you, Carl.
Hello, chemists. Today, I'm very humble and honored to discuss deuteration technology with such an esteemed group of chemists. As you know, deuteration is a process of replacing one hydrogen by a deuterium atoms in active ingredients. This exchange is a simple but interesting challenge to accomplish in a precise manner. So one question that I want to bring is, what is the rationale, the impact of deuteration in pharmaceutical development? There are several reasons. Having a deuterium on the molecule reduces the dosing, increased selectivity, reduced toxicity, and also increase the pharmacodynamic activity. When you put all this, it really shows that this hydrogen deuterium exchange is a very, very powerful tool to modify or improve the drug molecule pharmacological activities. A further proof of the value is the recent FDA approval for deuterated drugs for the use of in the use of genetics and autoimmune treatment. And also currently, other deuterated or active ingredients are being developed for and are at different clinical trial phases. When working with deuteration, there are different challenges that we have to overcome as chemists. One specific challenge that I want to discuss today is best way to control the site and extent of the percentage deuterium during the API deuteration. Different technologies, approaches has been developed for this incorporation. And one of those ones is the known hydrogen deuterium exchange approach. Due to the temperature requirements and conditions, the safety and tolerance to our functional groups and low selectivity can be challenges and difficult to overcome. And this is just one example. By using deuterated trifluoroacetic acid at 100 degrees, excellent yields. But notice that it deuterate every aromatic or exchange every aromatic hydrogen on the molecule. In many cases, that is not ideal to precisely add a deuterium into a molecule. A more attractive approach is the so-called deuterated pool synthesis that ensures the highest level of regio and chemoselectivity in the incorporation of deuterium into a molecule. And reductive deuteration is one of these pool synthesis approaches. In this case, using lithium and aluminum deuteride to precisely incorporate two deuteriums into molecules. This reductive deuteration is a census target. We want to provide the highest quality, the highest percentage deuterium during the reductive deuteration for life science applications. Let me move into the type of compounds we have been developing and our offering. There are three compounds: lithium aluminum deuteride, lithium triethyl borodeuteride, and sodium borodeuteride. As a chemist, these compounds are very valuable and offer three things: selectivity. They are very predictable in the number of deuterium added to the molecule. And also, they're very flexible during the synthesis sequence. So give a lot of option as chemists to where that deuterium should be, can be incorporated. The main differentiator between these compounds, it comes down to two things: the reducing power and the chemoselectivity that can be achieved with these reagents. I mentioned highest quality of our compounds. We use a validated NMR method that consists of reducing acetophenone and measure the percentage deuterium enrichment on the alcohol. And that enables us to offer these compounds with the highest quality of percentage of deuterium for reductive deuteration. Now that we have introduced these exciting or attractive compounds, let me provide more insights into the applications, the challenges, best approaches to use these reagents for organic synthesis. And if there is one concept to remember from this webinar, is a correlation from reactivity and selectivity. And it is a simple concept. A reagent more reactive is less selective. And the best way to compare reagents is, or relate, is one is compare an axe versus scalpel. The aluminum deuteride is the axe of reducing agent, and the other boron compounds are more of the scalpel to precisely surgically add the deuterium into molecules. Now, the balance of selectivity versus reactivity, especially with boron compounds, can depend and can be modified by the structure of the boron compounds, the counter ions, and additive. And also that can be done with aluminum, but especially with boron, is very valuable, this way to modify this compound. One last point that I want to emphasize or highlight: the number of effective deuteride transfer vary depending on the substrate and the reaction conditions used for the transformation. Let me go into a specific. And let me address reactivity of lithium and aluminum deuteride. And I explained before that aluminum deuteride is the axe of reducing it, is the most reactive one. And the table show it. It is very effective for reduction of amide to the corresponding primary amine. What the medicinal chemists wanted to achieve was the use of deuterium to modify the pharmacological activity of this DMT. And what was the problem with this, or what is the problem for treating depression disorder with this molecule? Short half-life and fast clearance. They obtain a very clean incorporation of 2 deuterium on the molecule. And this is what they found. They demonstrated longer shelf life, decreased clearance, but maintaining the similar pharmacological effects as DMT. So that's powerful, correct? Using a very reactive reagent to precisely add the deuterium and having the deuterium to modify the pharmacological impact of this type of molecules. My second example addresses or highlight the concept of reactivity versus selectivity. This case, the chemists wanted to reduce both functionality, the epoxy group and the carbonyl in one step. And they achieved that isolating over 56% and high incorporation of deuterium. They use this intermediate to build a more complex structure, more the geosmin deuterated analog. Now, it is a good reaction if chemoselectivity is not an issue, but it's not the optimal choice from reagent if the chemistry needs to be precise, that one of the 2 functionalities being reduced. And that's a concept that I want to expand in the next slides. The second consideration is deuterium transfer. And not surprisingly, the aluminum deuteride is also very effective for nitrile reduction, 94% to get this intermediate that was used for building this autoimmune disorder analog of compounds. And high degree of incorporation was obtained. Now, sometimes the challenge that we face as chemists with aluminum compounds is that the product can interact with aluminum, and that, as a result, require an excess to get the reaction to completion, excess of deuterium. That's inefficient in occasion, and that's something that can be addressed by other options for reductions with respect to reagents. And that is a great segue to lithium tritiborotodeuteride. And let me say a couple of things about this compound. This reagent can be as reactive, but also can be very selective. The first thing that I want to show is reactivity. As in the table, this SN2 displacement, it was a very clean, high inversion of configuration was observed, high deuterium incorporation, high overall conversion in the reaction. And also, very interestingly, only 3 equivalents of the reagents was used because it only had 1 deuterium to the molecule. Is a very efficient deuterium transfer for this type of applications. My second case is a very interesting and showcase the meaning of selectivities in organic transformation. The chemists, they wanted to selectively reduce the ester in the presence of a carboxylic acid. And the reason is that they wanted to then use a second step to introduce a methyl group to get a more complex structure. They achieved that with the tritiborotodeuteride. Tritibutylbutyrate, as seen at the table, it works great for ester, but for the carboxylic acid, it's an acid-base reaction, keeping the carboxylic acid intact for then the methyl lithium reaction to get the carbonyl the carbonyl group formation. The synthesis strategies, as you see here, is important. It's very important. Selecting the right reagent depending on the functionalities and chemoselectivity required for a transformation. Let me expand even further into selectivity. And this is a beautiful case where the monodeuteration was required. The chemists wanted to build this GABA analog. And the way they did it is by, as you see in the table, you require special conditions. By lowering the temperature, only one deuterium was added to the molecule, and that allows for allylation, hydroboration, and oxidation to generate or create these GABA analog compounds. Over 85% of results on the product. And again, it shows how selecting the reagent for chemoselective reaction is particularly a very important concept for consideration during the organic synthesis process. And that brings me to my last section of cases, the scalpel of reducing agents. Sodium borohydride. This is a very beautiful case, combining the cobalt. And I said before, an additive with sodium borohydride is a powerful combination. This cobalt sodium borohydride allows for the chemoselective nitro reduction followed by cyclization. It did not reduce it reduced the ester. Now, why this chemistry was so effective? The cobalt chloride is reduced with some borohydride to get this cobalt boride template. That allows for the close interaction between the reducing agent and the nitrile, and that is the reason why the so selective to get the amino and not reduce the ester. Now that's from the chemistry. What the medicinal chemist wanted was to understand the impact of deuterium on this intermediate or active ingredient for a COVID treatment. And they found over 40% improvements in the metabolic stabilization, on the shelf life, and as a result, lower doses requirements for to treat for COVID treatment. This example demonstrates how to use effectively a very selective reducing agent to introduce deuterium into a molecule to impact the pharmacological activity of a molecule. I said before that modifying the structure of the boron compound, the counterion and additive, will have a great impact in balancing this selectivity versus reactivity concept. Sodium borohydride and methanol by itself, without any other additive, is a very effective approach for ester reduction to give the primary alcohol. It involves high also incorporation. And notice also that some excess of the borohydride is needed. From borane, what is important to understand is that this solution needs to be stabilized, and this reaction some reaction will take place before then the reduction is possible. But overall, it's an excellent approach to get reduction of ester on the very mild conditions. My last two cases highlight how powerful these reagents are for deuterium incorporation. The olefin coupling is very difficult to do with reductive deuteration, as shown here in the table. Combining iron with boron deuteride enable free radical alkylation reaction in very high yields and very high percentage deuterium incorporation. It's a very selective. It tolerates several functional groups like alcohols, even carbonyl groups. And that's why it's a convenient, a powerful way to build more complex the structure using coupling of olefin molecules. My last example, I really like this case, and it's a very elegant approach to incorporate deuterium into aromatic rings. And essentially, it's a catalytic reaction to selectively deuterofluorinate at the ortho position of aromatic amides. And the reason why it works, and we can follow from the mechanistically, once the nickel inserts in the aromatic fluoride and is displaced, addition of deuterium reductively follows by then the addition to the aromatic. It's an irreversible process, and that's why the chemistry is so beautiful and under my conditions, irreversible. And that's where this type of approaches really open possibilities from synthetically to add deuterium and use this chemistry to build more complex aromatic structures. Before I move into the surveys, second survey, I hope I showed our offering. I showed the value of these reagents for reductive deuteration. And also I show what is challenges and how to overcome and why this chemistry is so powerful for deuterium incorporation in pharmacologic type of structures. I like now to give the floor to my esteemed colleague, Catherine. Catherine?
Thank you, Carl. We're just running a second survey now. You should see it on your screen. We're trying to get an understanding of your views on the potential impact of deuteration reagents in drug development. Can you please vote now for one of the following answer options? A, highly valuable. They offer clear advantages and are already influencing my work. B, promising. I see potential, but broader adoption is still emerging. C, uncertain. I'm not sure if they provide meaningful benefits over traditional approaches. Or D, limited. I don't believe they will have a significant impact in the industry. Just while the poll is running there, I just want to say to some people, they're asking about the link to this recording. Anyone who registered for this webinar will receive a link for this recorded webinar tomorrow, I believe. So, as discussed by Carl, deuterium can be strategically incorporated into drug molecules by reductive deuteration to create deuterated drugs. These compounds often exhibit improved stability and efficacy, potentially reducing side effects and enhancing therapeutic outcomes. Essence's deuterated reagents offer the following distinct benefits. 1, precise and selective deuterium incorporation. 2, predictable number of deuterium atom incorporation. And 3, flexible deuterium incorporation during the synthetic sequence. The application examples that Carl used highlight the fact that reactivity, selectivity, and deuteride transfer efficiency of deuterated reagents are critical for synthesizing deuterium-based APIs. Essence's specialties have a wealth of research in this area. Their current offering of lithium aluminium deuteride, lithium triethyl borodeuteride, and sodium borodeuteride are all manufactured in the US with a reliable global supply chain. They can offer grams to kilo quantities, but more importantly, they can provide their expertise in partnering with you on your deuterium journey, a partnership of innovation and expertise. So please do get in touch, tell us your challenge, and let us work with you to find a solution. In conclusion, deuterated reagents have emerged as a promising frontier in pharmaceutical research and drug development. Their unique properties and applications are reshaping the way we design, study, and optimize pharmaceutical compounds. As the industry continues to harness the potential of deuterium, we can expect to see safer and more effective drugs entering the market, offering new hope for patients worldwide. I would like to extend my thanks to Carl and Jenny from Ascensus, my own colleague Clodagh Phelan, for the work that went into this webinar today. And I'd also like to thank Specialty Chemicals magazine for hosting today's webinar. And lastly, thank you for taking the time out of your day to join us. We hope you will consider Comida and Ascensus as your partner of choice when considering drug deuteration applications.
Well, thank you, Carl and Catherine. That was absolutely brilliant. I see there's been a couple of questions pop up in the Q&A box there. So just to get you started, I'll read this one from Charlie Wise. What is the most effective way to advance deuteration in pharmaceutical development? And I'll hand that over to the experts there.
Catherine, you want to start, or should I give
you have the floor, Carl. You have the floor.
Yeah, from my point of view, one of the concepts of advancing this deuteration is commercial availability of these reagents. So we are putting a lot of efforts to have this compound on scale. The second component is that optimization of this. What is the reagent that will give the best approach, and that's something that we want to understand. We want to work with partner to be able to get this chemistry. So those are, I would say, the two areas where, as we as Ascensus want to partner and move forward with this technology into pharmacological development, we want to work together to get this into commercial application for reductive deuteration. Catherine?
Yeah, I just see there's another question there. What is the capacity of your production? So at the moment, as I said in the presentation, it's grams to kilograms. Isn't that right, Carl? But there is the possibility of scale up.
Yeah, we have right now not in the kilogram scale, but also depend on the reagents. For example, a commercial, and we are known well for this reducing A. And if you take a tritiboroduteride, this compound is in the, we can, because we have a commercial process for the hydride compound, then this is a reagent that we can rapidly put into multi-kilo scale. Then the other ones that we are at a developmental scale. But it depends on the need. We are 100% committed to make this type of reagents commercial, and we have the processes for these reagents to move it into scale, and depending on what is the specific application or what is the selection, and that's where we would like to work with you directly to understand what is the reagent that meets your need, and then enable us to rapidly commercialize and provide that to you for this application.
There's another question in there, Carl. Is there any future perspective to commercialize the lithium aluminium deuteride as a solution in a suitable organic solvent?
Yeah, this is an excellent question, and we as Ascensus, we want to provide the optimal solution to the customer. It is known, and this is the hydride version, where the aluminum compounds can be it's soluble in THF, it's soluble in ether, it's soluble in other solution like methyl THF. We have evaluated this option. Currently, from a customer point of view, the the powder is the one that is required. But if the solution, because of simplicity of adding the material to your reactor, is a better approach, we will work with you directly to develop that possibility. And that's what we are doing with in the past with other of our development, and that's our commitment to the customers.
Yeah, I think it's important to state there, this is a partnership approach that we're looking at. So we're looking at, partnering with our customers to find the right solution for them, a customized solution for them, for what will work in their process. There's another question in here, Carl. What is the deuteration degree in each of those deuterated reagents? What's the analytical technique used to determine the deuterium content? So I think you covered that on the slides.
Yeah, I covered that earlier in the slide. Is what we use. We validated NMR technique. It is a use test where we reduce acetophenone, and we determine the after reduction the percentage of deuterium in the molecule, and that directly indicates the percentage of deuterium enrichment that we achieve in our chemicals. From the validation, we have a high degree of guarantee or that this material will meet requirements of 98%. As you saw, this is our quality. We are 100% committed to safety and quality, and that's where we provide these validated methods and this material in the highest quality possible for this application.
There's another question there, Carl. Any other deuterated reagents for reduction, like any aluminium deuterioalkoxides?
Thanks for the question. The beauty of this reductive deuteration is it can be modified. You can modify the structure. We can modify the counter ion. We can modify the additive that we use. We at Sensus, we offer other reagents for reduction that include alkoxide. Specifically, we have not at the moment developed that for the aluminum deuteride, but we have the processes, we have the capability to provide the best solutions that give the right selectivity, the right reactivity for your application. And as I said, what we do is this partnership with our customer to satisfy his need and make the process a successful one when you go from lab to launch into your active ingredient.
Thanks, Carl. There's another question here just in relation to the NMR method. When you say validated, that's non-GMP analysis, isn't that right? Yes, that is the case.
That is the case. It's a non-validated GMP validated method for determining the percent of deuterium in our the compounds that we are offering for reductive deuteration.
Bear with me. What is the deuterium source for making these reagents? Is it D2 gas or D2O?
Yeah, it is not D2O. Let me put it this way. D2O will destroy any of this reductive deuteration. So that's we have a proprietary synthesis that is very effective, and that's why we are presenting today this webinar, and we are committed to commercialize this reagent for reductive deuteration in pharma core development.
All right, someone else asked here, what other deuterated reagents are offered by Comida Sensus? I mean, at the moment, Sensus has a very broad portfolio of reagents, and today I presented specifically reductive deuteration using for deuterium deuteride transfer. We have also catalysts that are very effective for deuteration using the deuterium gas, and also
we have a division for intermediate that enable us to support if needed for putting this incorporated deuterium. So we have many options that we would be like to discuss with you in more details to understand how can we support your advanced or your specific application for deuteration of organic molecules.
There's another question here. What is the most effective way to advance deuteration in pharmaceutical development?
Yeah, let me reflect on that. There are two things that, maybe three things that I want to answer. One is synthetically, if as a chemist you have this option to where you want to put the deuterium, you have a conversion process of, or you are late on the synthesis. Having option a toolbox of reagents that enable you to selectively, to surgically, to precisely add the deuterium where you need it is very important. The second part that is more on the pharmaceutical, when they are evaluating where that deuterium will be in the molecule, we play a role, and that's why we want to have as broad portfolio as possible to help the chemist to add the deuterium where it needs to be. And the last point is, and that's why we are discussing this topic today, is the commercialization of these compounds. And we have a very long history of with these reducing agents, the hydride, and we have our belief is we have the capability to support you from lab to launch. And those two things will be what I consider as two key aspects to commercialize, to advance deuterium into pharmacore development.
Thanks, Carl. Another question here. Are there specific chemistries or structures that present challenges for the deuterium incorporation?
Yeah, that's I explained. I showed different examples today, and it comes down to this one concept that I want you to remember from this webinar: the selectivity versus reactivity. Some molecules have multiple functional groups. You have an ester, you have an amide, and especially if you need to just reduce that functional group, the selection of the reagents become critical. And sometimes the reaction conditions will allow you put in the additive to do it, but in some other cases, the more the axe is required to get this for bulky functional groups, and that's where that as a chemist needs to have doing the evaluation to decide if the deuteration will work or not, and to do that reduction. So those is a systematic approach, and I know that with this STEAM group of chemists that's where the bulk and the beauty of having options for deuteration, having options for selective, precisely adding the deuterium into the molecule.
Thanks, Carl. We got another question there again. It's about the lithium aluminium deuteride. Is it available in THF? So I think you did cover that already. That's certainly an avenue you can look to with a customer. You can provide them with a customized solution if that's what they need, if that's what they need to feed their reactor. Yeah,
and I would love to talk to this chemist and understand his application and the use of the solution. Is again also selecting the reagent. For example, lithium deuteride is in solution. We can make this aluminum deuteride in solution to facilitate from an engineer point of view to transfer these compounds into a reactor and more commercial scale. So yes, it is an option. I would love to discuss that further. Your need and come up with a customized best solution for your application.
Thanks, Carl. That person has actually left their email address, so we'll certainly be able to reach out to them after the webinar. So thank you for that. Question in here about the regulatory landscape. What does the regulatory landscape look like for these deuteration reagents, especially at large scale, potentially greater than 1 ton, if that is possible? Is this one reason for having the production in the US?
That is I would like to, I have to go back to you on that one. And specifically from regulation, and let me go from a customer, and I'm not into pharmaceutical for all the regulatory, but my understanding is that the deuterium isoter of hydrogen is a way that really significantly reduce any issues from regulatory, because deuterium versus hydrogen, the difference is 1 neutron, and that's makes really facilitate the advancement of these pharmaceutical intermediates into development. So I do not have all the information today for regulatory. I'd like to go back to you in terms of our reagents and also from a pharmacological. But I would say deuterium from isoter is probably the best one to minimize any regulatory issue as you're moving this active ingredient into commercialization.
Carl, there's a question here. What is the safety of the lithium aluminium deuteride versus lithium aluminium hydride?
Yeah, the safety, and I said before, we live and die with safety, and then that's we have a product stewardship, extensive product stewardship to ensure that any chemist that use our reagent will do it safely, and then can then advance it to commercialization. Let me discuss the 3 compounds and the difference with the 3 compounds. The lithium aluminum deuteride is the most reactive. It's a flammable solid. So there's from handling, there's some consideration that we will be more than happy to specifically discuss with our product stewardship and actually go to your plant to help you to ensure that it is handled as safety as a powder. The solutions of tritiborodeuteride is in a cylinder, so it's as long as you pipe it properly, and then there's no leak, then that minimizes the interaction of the operator with the material when having a tritiborodeuteride as a solution. That brings me to the borodeuteride. The borodeuteride can be handled and for a period of time when transferring that to a reactor. From a flammability is a safer option, easy to handle. But again, I would say the 3 compounds has different safety and handling consideration that we will be specific, and that's also our commitment, our offering, that if you use any of our reagents, we will provide extensive safely and handled of this reagent to operate safely at your plant.
Yeah, and I can second that. I've been at some of the customer meetings with the Census where they've done the safety and handling training, and they are absolutely comprehensive and complete and excellent. They take the questions, they have all the information on the best way to handle the products. I mean, to be honest, with a Census, you get a full partnership here. You know, they'll work with you to find the best solution for you, and then they'll work with you to ensure you use that safely at the site. And again, the regulatory backup will be there as well, and the support. And I can safely say that, having worked with them for many years. I think we have dealt with all of the questions at this stage. If I'm missing something, please do type in now. It's great to have so much interaction on this webinar. Carl and I are both very appreciative of that. And it was obvious from the polls that the people on this webinar, quite a lot of them are familiar with deuteration and are using it currently. And really, they are the people we want to be talking to. So we do appreciate you coming on today and getting involved, answering the surveys, sending in your questions. We will follow up with anybody who's left an email address You'll be sent the link to the webinar, I believe, tomorrow. And as well as that, if there's anything else that comes to your mind after the webinar, please do just email info@camida.com, CAMIDA, and I will get back to you, or I will forward your query on to Carl, and Carl will get back to you. But between the two of us, we'll have you sorted. Oh, hang on. There's just one. Oh, somebody just said thank you very much.
Yeah, and to finalize, to finish this webinar, again, it was a real honor for me to discuss deuteration technology for pharmacore development, and I was very excited about from the question all the possibilities and how we can really partner from lab to launch to advance your application into commercial scale. Thanks again for considering us as your partner, as your supplier choice for this reagent, and I'm looking forward to discuss this in more details in a future occasion.
I think that's us finished for today then, Carl. We'll sign off now, but thank you all once again for taking the time to attend this webinar, and please do reach out if there's anything you want to follow up on, info@camida.com. Thank you and enjoy the rest of your day.